इनका ही आवाज ज्यादा आएगा होगा। जोर से बोलो। ठीक है, ओके। काम कर रहा है ना कीबोर्ड के बाद, स्टार्ट करना ना इसके बाद। करो स्टार्ट। Good morning to all of you. Today we will discuss two topics. One, in continuation of the last talk which I had said about the gastric secretions and the gastric regulations. And then once we complete with the gastric motility, we will start with the functions of the pancreas. We shall deal with the exocrine functions of the pancreas. Now, if you just go back to the last class and recollect or recall what we had discussed, we had talked about the gastric secretions, the regulation of the gastric secretions, and the factors which are affecting or protecting the gastric mucosa from the hydrogen ions. One of the other important functions apart from the gastric function is the storage function. It stores the food in the stomach so that the food gets digested. When it is digested to a partial extent, the stomach has to send the food or the bolus or the chyme into the small intestine, that is the duodenum, for further digestion of the carbohydrates, proteins and fats to take place. We also know that the storage time or the gastric emptying time is around 3 hours. On one hand, the food which is present in the stomach is getting digested and trying to slowly push the contents from the stomach into the duodenum. On the other hand, the duodenum is receiving the food from the stomach and trying to inhibit or stop the gastric contractions or the gastric emptying so that a balance is maintained. Now let us very briefly discuss what are these two factors which are promoting the gastric emptying and what are the other factors which are inhibit the gastric emptying. One of the most important stimulus for the emptying of the stomach is the presence of the food in the stomach. As soon as the food which we eat enters into the stomach contraction starts and the mixing of the food and the propulsion of the food towards lower down of the stomach happens. So now what are the causes which causes these contractions of the stomach to take place? First, the presence of food inside itself will create a small pressure or increase the intragastric pressure and cause the movement of the stomach wall. But this factor is to a lesser extent responsible for the gastric emptying. The main factor which plays an important role in the gastric emptying is the stretch of the stomach wall. We know that as the quantity of the food which we eat goes on increasing, the stomach wall gets more and more stretched. We know that the stomach wall is made up of smooth muscles. There are circulatory muscles and the longitudinal muscles. These muscles get stretched up. When they get stretched up, the smooth muscle wall gets stretched up, and when the smooth muscle wall gets stretched up, it initiates contraction because the property of the smooth muscle is contractility. So stretching up of the gastric wall initiates these propulsive movements and the stomach contractions of the gastric contraction starts and gastric emptying takes place. The other factor which plays an important role in the gastric emptying is the presence of gastrin. Whenever food enters into the stomach, it stimulates the G cells which are present in the cardiac end of the stomach and the body and the fundus of the stomach. These G cells secrete gastrin. Though the one of the most important functions of the G cells is to stimulate the secretion of hydrogen ions, but it also has got a role on the circulatory muscles of the gastric wall and causes the contractions of the stomach. So in other words, the gastrin hormone is functioning in two different ways. One, on the secretion side, it is causing the parietal cells to secrete more amounts of hydrogen ions. On the other hand, it is acting on the circular muscles or the smooth muscles of the stomach and causes contractions. So basically, there are two factors which promote gastric emptying. One, the presence of food in the stomach causing stretching of the gastric muscle, gastric wall. Second, the release of gastrin from the G cells will initiate smooth muscle contraction. Now, once the gastric muscle starts contracting or the stomach emptying starts, it pushes the content or empties these contents into the small intestine through the pylorus. Now, the pylorus opens up, allows the digested food or the partially digested chyme to enter into the duodenum. Once the food enters into the duodenum initiates certain reflexes because of which the gastric emptying is reduced or the gastric contractions are reduced. So we have a situation where the gastric wall or the stomach is emptying its contents into the duodenum. The duodenum in turn on receiving the signals stops the gastric contraction. Now, what are the factors which initiates or triggers these things? First, as soon as the food enters into the duodenal wall stretches. Stretching of the duodenal wall will stimulate the enterogastric reflexes. These reflexes in turn will go into the stomach, and on the stomach wall they will inhibit the contractions of the gastric circulatory muscles. In this way, one of the mechanisms by which duodenum prevents the gastric emptying is through stimulating the or initiating the local myenteric reflexes. The second factor is the presence of any irritants in the gastric mucosa, in the duodenal mucosa. As chyme is entering into the duodenum, the osmolarity of the duodenum, instead of being a normo-osmolar solution, it may be a hypertonic or a hypotonic solution. So this presence of hyperosmolarity or hypotonicity of the chyme will cause the duodenum to initiate the local myenteric nerves to inhibit the gastric emptying. Third, the food or the chyme which is entering from the stomach is in fact acidic in nature. The presence of acid chyme in the duodenum immediately inhibits the gastric emptying. Fourth, the presence of any irritant substances in the duodenum or presence of these partially digested proteins and fats in the duodenum will inhibit the gastric emptying. Once this gastric emptying stops, the pylorus closes and further entry of the chyme from the stomach into the duodenum gets stopped, so that sufficient time is there for the chyme to remain in the stomach so that further digestion takes place and the duodenum receives only small quantity of chyme which is able to digest. So the other factor which inhibits the gastric emptying is the presence of excess chyme in the duodenum. If there is a sudden rush of contents from the stomach into the duodenum, this excess of chyme in the duodenum will immediately inhibit the gastric emptying. So these are the factors which will inhibit the gastric emptying. So on one side we have got factors which will promote gastric emptying, which are per se present in the stomach itself. On the other hand, we have got the duodenum where it inhibits gastric emptying, and a balance has to be maintained, and what in fact decides whether the stomach gastric emptying should happen or not is the rate of flow of chyme from the stomach into the duodenum. It is at an optimal level. Sufficient rate of fluid, sufficient rate of chyme is coming from the stomach to the duodenum, so that the duodenum can digest the incoming chyme, the partially digested proteins which are coming. Then the gastric emptying takes place at a normal phase. But if there is an excessive rush of fluid coming, of chyme coming from the stomach into the intestine, immediately the gastric emptying is stopped, so that sufficient time is present within the duodenum for the already received food to get digested and broken down into simpler substance. Now we shall now go on into the pancreas. We shall see now the functions of the pancreas, the exocrine functions of the pancreas. The pancreas is a compound gland which is structure similar to the salivary glands. A compound gland means it has got definitely branches where it is divided into. The gland is not a single gland. It is divided into number of lobes. Lobes are multiple. Lobes are broken down into further lobules, lobules into further lobules, smaller lobules, and ultimately we have got small acini. So basically, the whole of the pancreas consists of two basic structures. One, the acinar cells or the acini, and the other is the ducts. Here, two different structures which are forming the main basis of the pancreas have got two exclusive functions. The acini are responsible for the secretion of digestive enzymes, whereas these ducts are responsible for secreting bicarbonate only. Now I will briefly describe to you the structure of the acinar cells and the ductal cells. So here if you see, there is a organization of the pancreas. If you see the diagram there, here it is the main pancreatic duct. This pancreatic duct combines with the common bile duct, and these two open into the duodenum. Now this is the main pancreatic duct. This pancreatic duct is broken down into number of ducts, which we'll call as the interlobular ducts. So these are the interlobular ducts. The interlobular ducts are further broken down into intralobular ducts, and these interlobular ducts further break down into an intercalated ducts. To see the structure of the gland, the acinar glands, we have as a pancreatic glands, we will find here that this pancreas is broken down into number of lobes. Each lobe has got a main duct which is called as a lobar duct. Number of lobes form the intralobular duct. The intralobular duct form an interlobular duct. Number of interlobular duct opens into the main pancreatic duct. Now each of these lobe is further divided into a number of acini. You see these are the acinus. So number of acini are present, and each of these acini are lined by certain epithelial cells which are known as the acinar cells. So this is the acinar cell here, where you will find that there are the intracellular structures, various intracellular structures present, where you have got the rough endoplasmic reticulum, the mitochondria, the Golgi apparatus, and the other organelles present. All these organelles together perform one main function within the acinar cells, is the secretion of the enzymes responsible for the digestion of carbohydrates, proteins, and fats. These enzymes, which are synthesized within the acinar cells here, are stored in the zymogen granules. These zymogen granules are the secretory granules, and these granules contain the enzymes which are responsible for the digestion of the food substances, and they are basically present in an inactive form. So remember one thing, that the function of the acinar cells is to secrete only enzymes, digestive enzymes. As we see the histology of the ducts, whether it is the interlobular duct or it is the intralobular duct or it is the acinar ducts, we will find that the epithelial cells of these is responsible only for secretion of the bicarbonate and chloride ions. In other words, the gland cells, which are the acinar cells, are responsible for the synthesis, storage of the pancreatic enzymes, whereas the ductal cells, which are the cells which are lining the ducts, are responsible for the secretion of the bicarbonate ions. The next few slides I'll explain to you what is the importance of these things. The same thing again has been explained here. You have the acini, the acinar ducts, you have got the intercalated ducts, these are forming. Then you have got the intralobular ducts, forming the extralobular ducts, extralobular ducts forming the main collecting ducts. So as these acinar cells synthesize the zymogen granules, they secrete this into the ducts as a pancreatic juice. So the composition of the pancreatic juice secreted by purely the acinar cells is only enzymatic in nature. So as this passes, pancreatic juice with the enzymes passes through the ducts, the epithelial cells lining these ducts, or the ductal epithelial cells, will go on secreting fluid which is rich in water and bicarbonate ions. Thus, by the time this pancreatic juice enters into the pancreatic duct, it is containing two components, one the enzymatic component or the solid component, and the other is the electrolyte component, where it is rich in bicarbonate ions and water. This is further explaining this, where you will see that here it is the acinar cells. In the acinar cells there is a spontaneous secretions, where there is more of the enzymatic side, but very little amount of bicarbonate is secreted. But as this this pancreatic duct comes out of the acinar ducts, goes into the intralobular ducts, you will find there is a rich secretion or a increased secretion of bicarbonate ions into the lumen of the duct. So ultimately the pancreatic juice which comes out from which is secreted into the lumen of the duodenum consists of two components. One, a fluid component which is rich in bicarbonate ions with water, and the other is the enzymatic component consisting of digestive enzymes. Now, as this food enters from the stomach into the duodenum, or as chyme enters into the small intestine, there has to be two important things have to take place. We know that the food, when it is in the stomach, is having an acidic pH. There is a gastric secretions or a gastric juice which is having high quantities of hydrogen ions. So as this gastric secretion or this chyme enters from the stomach into the duodenum, it is important that the acid pH must be neutralized in the duodenum. Otherwise, presence of acid in the duodenum will result in damage to the duodenal mucosa and ultimately result in ulcers. This is one of the pathophysiological bases for all duodenal ulcers. If there is an excess hydrogen ions entering from the pylorus or from the stomach into the duodenum, and the duodenal cells are not able to neutralize that acid content, that hydrogen ions, it results in the desquamation of the mucosa of the duodenum, thus resulting that mucosal barrier is broken down. If this mucosal barrier is broken down, it results in ulceration of mucosa. Ultimately, it will result in pain, and it is called as duodenal ulcers. If it is left untreated, it may cause perforation and complications, which you will learn later as you go into your medicine and surgical postings. But basically, you have to remember here that as chyme enters from the stomach into the duodenum, it has to be neutralized. The other function here is in the small intestine, the food which has been broken down into simpler substances but only partially broken down has to be completely broken down. That is, the proteins, carbohydrates and the fats has to be broken down into the most simplest substances, so that in the simpler form absorption of the food can take place. So how does pancreas play a role here? The food is present in the duodenum. The pancreas is a present retroperitoneal organ. What is the role of the pancreas here? Pancreas plays an important role through its secretion by pancreatic juice. And what does this pancreatic juice do? Does two things. One, it secretes digestive enzymes for digestion of all the foods, all the types of foods which are present in the chyme. And secondly, it secretes pancreatic juice rich in bicarbonate ion so that it neutralizes the acid chyme. So let's briefly discuss about what is this protein digestion. In this pancreatic juice, the enzymatic part consists of certain proteolytic enzymes. These proteolytic enzymes consist of trypsin, helps to break down trypsin, consist of trypsin, chymotrypsin and carboxypeptidases. The trypsin and the chymotrypsin breaks down the proteins into polypeptides. Carboxypeptidases will break down the polypeptides further into amino acids. Then it also consists of certain enzymes which are responsible for the carbohydrate digestion. Basically, it is called as pancreatic amylase. This amylase is responsible for the breaking down of starch and glycogen to disaccharides. Remember this enzyme, pancreatic amylase. This is an important enzyme which helps in the carbohydrate digestion. It is also clinically important because in certain clinical conditions where the pancreas is injured or pancreatic infection occurs, where there is inflammation of the pancreas, which we call it as pancreatitis, this pancreatic amylase is liberated freely into the circulation, and if you measure the pancreatic amylase, it gives you an idea of the extent of inflammation in pancreatitis. So one of the marker for acute pancreatitis or chronic pancreatitis is the measurement of the serum amylase. If the normal serum amylase is more than 2 times to 3 times, it indicates that there is a severe inflammation of the pancreas. The pancreatic acinar cells are liberating the enzymes into the bloodstream, and this is the marker to know the progress of the disease, that is pancreatitis. But for now, just remember that the carbohydrate digestion takes place because of the presence of amylase in the pancreatic juice, which breaks down starch and glycogen to disaccharides. Next, it also has certain enzymes which are responsible for the digestion of fat. Mainly the enzymes are the pancreatic lipase, the phospholipase and the cholesterol esterases. The pancreatic lipase breaks down the fats into fatty acids and monoglycerides. The phospholipases breaks down the phospholipids into simple fatty acids, and the cholesterol esterases also breaks down the cholesterol esters into fatty acids. Clinical importance, remember here, the pancreatic lipase is another enzyme where the amount is raised in pancreatitis. So measurement of the levels of the serum lipase in pancreatitis, either acute or chronic, will help you to know the progress of the disease and the extent of the damage has taken place. Now the next question is, if the pancreas consist of enzymes which are digesting proteins, fats and carbohydrates, why doesn't the enzymes destroy the pancreas gland itself? For this, one thing you should remember here is all these enzymes which we have discussed, either the carbohydrate pancreatic lipase or the pancreatic esterases or the proteases, are all present in an inactive form. They are stored in the zymogen granules in the inactive form. They get converted into the active form only once they are in the duodenal lumen. As long as they are in the pancreatic gland itself, stored in the zymogen granules or passing through the ductal cells, they all are in the inactive form. In addition to this, there is also a substance present which is known as a trypsin inhibitor. This trypsin inhibitor is to it that these enzymes do not get activated. So trypsinogen, the enzymes which are present there are in the inactive form in the form of trypsinogen, chymotrypsinogen and procarboxypeptidases. For these enzymes to act, this has to be converted from an inactive stage to an active stage. That is, trypsinogen must get converted into trypsin, chymotrypsinogen has to get converted into chymotrypsin, and pro-carboxypeptidases has to get converted into carboxypeptidases. Now, how does this happen? This conversion of all these inactive enzymes into an active structure happens in the presence of an enzyme which is known as enteropeptidases. This enteropeptidases ensures that the enzymes are converted from the proactive form or the inactive form into the active form. Now, what does this enterokinase does? This enterokinase acts on trypsinogen and gets converted into trypsin. And where is this enterokinase present? This enterokinase is present on the intestinal mucosa cells. Remember here that it is present on the intestinal mucosa cells. It is not present within the pancreatic gland or in the ductal cells. So once the pancreatic juice enters into the duodenum, that is the posterior aspect of the duodenal wall where it opens into the duodenum, there these cells are secreting enterokinase. This enterokinase activates or converts trypsinogen into trypsin. Once trypsin is getting active, it autocatalytic activation takes place, where it means that a small amount of active trypsin will convert inactive trypsinogen into trypsin on its own. It also activates, this trypsin also activates chymotrypsinogen, pro-carboxypeptidases, and trypsin. So basically, inactive forms of enzymes are present. The inactive forms is maintained by also a trypsin inhibitor, so that if any trypsin is present within the pancreatic gland, it doesn't get activated. Once the pancreatic juice enters into the second part of the duodenum, there the enterokinase is secreted. This enterokinase converts a small amount of trypsinogen into trypsin. Once the trypsin is getting activated, it goes on an autocatalysis, where it itself activates the inactive enzymes into an active forms. So where are these trypsin inhibitors present? The trypsin inhibitor is present in the acini and in the ducts. Then what is acute pancreatitis? Now, whenever an acute pancreatitis means sudden inflammation of the pancreatic cells. What are the pancreatic cells which get inflamed? Two types of cells which are present. One is the ductal cells and the other is the acinar cells. So whenever there is an inflammation of these cells, that means some activity has taken place, and this happens because either there is total lack of trypsin inhibitor, or the trypsin inhibitor is in less quantity than what is required. So less quantity of trypsin inhibitor is not able to inhibit a large amount of trypsinogen to trypsin. Hence there is a conversion of trypsinogen to trypsin happening within the pancreatic duct, within the pancreatic gland. So this pancreas gets inflamed or gets swollen up, and more quantities of enzymes are released into the circulation. That condition is called as acute pancreatitis. Acute condition means it lasts from 1 week to 2 weeks or 3 weeks. If this condition persists for a long period of time, then it is called as a chronic pancreatitis, or in other words, later on there can be a acute or chronic pancreatitis. Now basically, coming back to the physiological process here, let us see how this bicarbonate neutralizes acid chyme. One of the components of the pancreatic juice is high quantities of bicarbonate present. The function of this bicarbonate is to neutralize the chyme which is coming into the duodenum from the stomach. How does it do that? First of all, one hormone which is responsible for inducing this bicarbonate secretion is secretin. So under the influence of secretin, bicarbonate secretion increases. Once the bicarbonate secretion increases, as you see in the diagram here, hydrochloric acid is coming from the chyme into the duodenum. In the duodenum, the pancreatic juice is present. This pancreatic juice consists rich amounts of bicarbonate, that is sodium bicarbonate. So here you will find that the sodium chloride gets converted, and the carbon dioxide and plus this carbonic acid is formed. This carbonic acid breaks down into carbon dioxide and water. So in this way, what acid hydrogen ions are entering into the duodenum are getting neutralized by the presence of these bicarbonate ions. So what is secretin in fact doing? Secretin is a hormone which is called as the nature's antacid, because the moment there is a acidity in the stomach or a hydrogen ions are entering from the stomach into the duodenum, the nature of the body is causing the secretin to be released. Once secretin hormone is released, it facilitates increased secretion of bicarbonate rich pancreatic juice, and this bicarbonate neutralizes the hydrogen ions. Now this is the model of the bicarbonate secretion. Here carbon dioxide combines with water in the presence of carbonic anhydrase in the cell. Carbonic anhydrase dissociates into a hydrogen ions and bicarbonate. Hydrogen ions are transported out off through the sodium gradient mechanism, whereas bicarbonate moves out in exchange for chloride ions. Now the rate of hydrogen ion bicarbonate ion secretion is totally dependent upon the chloride concentration. It moves against the gradient here, and one cell, one hormone which is responsible here is secretin. The same thing which is being shown here. Here you will find that there is a ductal cell. This is the lumen, which is the pancreatic juice, which is the pancreatic duct. Here on one side you have got the blood. So here you will see that this carbon dioxide and water is there, bicarbonate is here. So you will find that the bicarbonate is moving out, chloride is moving in. So in this way, the hydrogen ions and the bicarbonate, that is H2CO3, gets broken down into hydrogen ion and a bicarbonate ion. What happens to the hydrogen ion? The hydrogen ion goes out from the ductal cells into the blood, where it is again combining with the hydrogen ions with the bicarbonate present here to form carbon dioxide and water, whereas this bicarbonate ion is secreted into the lumen, and when it is secreted into the lumen, chloride ion enters it. This is the chloride shift which also happens here. So in this way, now this bicarbonate which is present in the lumen will combine with the hydrogen ions which have come into this lumen from the stomach by the chyme, and that will get neutralized. So in this way it is said that the hydrogen ions are neutralized by bicarbonate, and this whole movement of bicarbonate and chloride in opposite direction, and this excess secretion of bicarbonate ion within the ductal cell is facilitated by the hormone secretin. Now what is the effect of secretion rate on the ionic composition of pancreatic juice? Simple. If there is a low secretion rate, that means the bicarbonate concentration is low. So when there is a less amount of bicarbonate, less amount of pancreatic juice secreted, the bicarbonate concentration is low and the chloride concentration is high. When there is a rich or a more quantity of bicarbonate ions being secreted, pancreatic juice secreted, it will have a high concentration of bicarbonate and low concentration of chloride. So let us see how is the pancreatic juice regulated. Similar to the gastric juice secretion, we have got a cephalic phase, we have got a gastric phase and intestinal phase. So also here are the phases, but its regulation is by means of two hormones. The two hormones which are playing an important role here are the secretin and the cholecystokinin. In addition to these two, the local reflexes, that is acetylcholine through the vagus nerves, also plays an important role in the secretion of the pancreatic juice. Let us basically see from this diagram, let us understand what happens. Now as this food is entering from the, as this chyme is entering from the stomach into the duodenum is getting stretched. The stretching of the duodenum will cause the stimulation of the local myenteric reflexes, and this local myenteric reflexes will stimulate the pancreas to release certain enzymes into the acinine. Secondly, as acid is entering from this, as this chyme is entering from the stomach into the duodenum, the partially digested proteins and fats will, and certain amount of hydrogen ions which are present here, will stimulate the cells which are present lining the lumen here, and will cause the cells to secrete secretin and cholecystokinin. This secretin and cholecystokinin enters into the circulation, and from here it enters into the pancreas, and in the pancreas it stimulates the pancreatic ductal cells and the acinar cells to increase the secretion. The secretin causes increased secretion of pancreatic juice rich in bicarbonate. The cholecystokinin causes secretion or stimulates the acinar cell to cause secretion of the enzymatic part of the pancreatic juice. Acetylcholine, which is released through the myoventric plexus, will stimulate both the acinar cells and the ductal cells, and basically it is the stimulating the ductal cells to cause increased secretion of bicarbonate ions. So secretion of fluid and bicarbonate is mostly dependent upon the hydrogen ions which are entering into the duodenum. Secretion of enzymes is mainly dependent upon the fat and the protein which is entering into the duodenum. Here is a diagrammatic representation of the role of the hormones. So two important hormones are playing a role here. One, we see that as food is entering into the duodenum, here you will find that there are certain cells which are known as the S cells in the duodenal mucosa. These S cells will get stimulated because of the presence of food and presence specifically of the hydrogen ions. These hydrogen ions will cause the S cells to get stimulated. The S cells will be released from here, will release secretin hormone. The secretin hormone will enter the circulation, reach the pancreatic gland. In the pancreatic gland, it will stimulate the ductal cells to cause increased secretion of water and bicarbonate. This water and bicarbonate, which is the blue line, will pass through the pancreatic duct and opens into the second part of the duodenum, and here in the second part of the duodenum, as the hydrogen ions are coming down, the bicarbonate ions will interact with it. So the hydrogen ions with the bicarbonate ions will get converted into water and carbon dioxide, and in this way the hydrogen ions will be neutralized. Further down, as the chyme is passing further down into the duodenum, there are certain cells which are known as the I cells. These I cells will stimulate the release of cholecystokinin. It's another GI hormone. This cholecystokinin will be released from the I cells of the duodenal mucosa, enter into the circulation. Once it enters into the circulation, it then acts on the pancreas. It acts on the acinar cells of the pancreas and causes a secretion of pancreatic juice which is rich in the enzymes. Specifically, these are the proenzymes or the inactive enzymes which can digest, consisting of proteins, carbohydrates and fats, and these acinar secretions which are rich in inactive enzymes pass through the pancreatic duct, open into the duodenum, and once they are in the duodenum, here you will have got certain mucosal cells which will secrete enterokinase. This enterokinase will act on this chyme or the enzymes which are present here in the orange line, and they will activate the conversion of trypsinogen into trypsin. Once this trypsinogen gets converted into trypsin, it will convert all the inactive enzymes into the active enzymes. Once the inactive enzymes are converted into active enzymes, here the digestion of the partially digested proteins, fats and carbohydrates takes place, and as this food moves down the duodenum into the small intestine, complete breakdown of the food substances into smaller products, into simpler products takes place, so that in the lower part of the small intestine they can get absorbed into the circulation and carried in the circulation in the form of carbohydrates, that is glucose, amino acid and simple fatty acids. Now when is secretin is released from the S cells only when the pH is less than 4.5. Maximum secretion of secretin occurs when the pH is less than 3. So here again it's a diagram which is showing the various receptors and how it is acting. These are the first messengers, that is the cholecystokinin, gastrin. Here it is a secretin, then the cholecystokinin, secretin, which are basically secreted. These are the first messengers. They bind here and they inactivate. They bind onto the receptors which are present on the pancreatic cells. Maybe this is the ductal cell or the acinar cells. Once this binding takes place, they activate the second messenger. The second messenger can be a cyclic AMP, and in case of cholecystokinin, it is the IP3 and the DAG. So secretin basically acts through the second messenger through activating cyclic AMP. Cholecystokinin acts by activating the DAG and the IP3. Once they act on the second messengers, they act on the third messengers to increase the phosphokinase and calciuminins and phosphokinase. Once the third messengers are activated, they cause increased secretions of bicarbonate ions if it is a ductal cell, and they cause increased secretion of enzymatic portion if it is an SNR cell. Phases of gastric pancreatic secretion. Again, as I said earlier, there are three phases. We have got the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase is the phase where the sight of the smell, sight of the food, the thought of the food, the smell of the food, or the taste of the food itself will cause the release of small quantities of pancreatic juice secretion in the duodenum, and this accounts for around 20%. The gastric phase, which is the phase where the pancreatic juice is secreted when the food is in the stomach, is called the gastric phase of pancreatic secretion. It is just about 5 to 10%, whereas the intestinal phase is that phase of the pancreatic secretion where the pancreatic secretion is increased tremendously when the food is in fact present in the intestine. The stimulus which is causing this is the presence of acid, fat, proteins, and carbohydrates there. So around 70 to 80% of the total 24 hours pancreatic juice secretion takes place when it is in the intestine, 5 to 10% in the gastric phase, 20% in the cephalic phase. With this, we complete the pancreatic juice secretions. Thank you, one and all. Have a nice day.